welcome to the final revision stream for AS levels. I honestly cannot believe how quickly the time has flown by. I literally can remember, like, it feels like a couple of weeks ago when I was sat here, saying, alright guys, it's exams in a couple of months, but we're almost done. One more day of exams, and then we're on the clear. We've gone clear. So what I want to do in today's stream is literally go through as much as possible, and I promise I'll make this worth you guys' while. What we're going to be looking at are the extended response questions, like the 4-5 markers, where they want you to ramble about a specific thing in detail. Like, mainly like the processes, for example. Like, if you got a 6-mark question on lipid digestion, how confident would you be that you would be able to tick every mark scheme point? Because I'm pretty sure I wouldn't be able to, so I don't know about you guys. So that's what we're going to be looking at today. We're going to go over a couple processes which I can almost guarantee are going to come up in this exam, and we'll just go from there. We'll go from there. So before we crack on, I do actually want to send the stream link to a couple of people. I the thing is, I really doubt people watch these, except for like the 300 people which somehow ended up watching the last one. So I don't know if sending the link to anyone will achieve anything, but we'll do it nonetheless, because like, if I can help someone out, or they can help me out, that's a win. Apologies if you guys can hear my neighbours in the background. We have brand new neighbours, and I don't know where they're from or anything, but this guy who must be in like the literally right behind me at this point, he's been talking on the phone for like 2 hours, and I don't know a thing he's saying, so apologies if you can hear random like shouting. That is not me or my family. Do I even have WhatsApp on my computer? I bet I don't, do I? I bet I don't. Okay, my apologies for for that. Right, let's get on with it, shall we? Let's get on with it. So again, no idea if anyone's gonna watch, but we'll crack on nonetheless. So the first thing I want to go over is arguably like the easiest thing. I think it's the thing that we know the most about, protein synthesis. Now, I'll be completely honest with you guys, and this is proof that revision is actually decent. I'll be completely real with you. When we did that lesson on transcription and translation, I swear down, I literally was walking home with Ellis, and I was like, bro, I have no idea what any of that lesson meant. I have no idea what any of it means. And it took me about an hour of just non-stop revision to finally understand what it was, about all of the different things about protein synthesis. But now I would argue that I know it very well. So again, proof that literally an hour of revision can completely change a subject. So let's start with transcription. And before we can actually start rambling about it, we should probably like draw it just to get like a nice little visual. So assume that this is like our DNA here. Okay. So the first thing which happens is that the hydrogen bonds between the bases on both strands of the DNA are broken. Now, you cannot get a mark for mentioning hydrolysis here, because I'm pretty sure hydrolysis isn't involved. You might not even get a mark if you mention the enzyme responsible for this, so don't risk it. All you need to say is, for 1 mark, hydrogen bonds, don't put H bonds, but hydrogen bonds, broken between bases. And the effect of this is that you're going to have 2 strands now. 2 strands, because the DNA molecule unwinds. And what you have is one of these strands is going to act as a template for RNA. Okay, one of them. That's how it's different from DNA replication. So only one strand of DNA acts as template. Now what you have, and this bit took me a while to process, but I think I understand it now, is around the DNAs, you have these little free-floating nucleotides, RNA nucleotides. And what happens is these free-floating RNA nucleotides are going to bind, they are going to align to their complementary base. So, free-floating RNA nucleotides align to complementary base, or you could just put complementary base pairing. Next, you need to mention the whole idea of phosphodiester bonds join adjacent nucleotides. Phosphodiester bonds join adjacent nucleotides via the action of RNA polymerase. And I don't know if you get marks for mentioning condensation, but that is what happens. And finally, you need to say splicing of pre-mRNA. So yeah, what you now have is, remember, hydrogen bonds don't form because this is a copy which is meant to move. So you now have this RNA copy which is going to move out of a nucleus. But before it can do that, remember the introns need to be removed. And that's basically how you would get 5 marks, I'm pretty sure. So the hydrogen bond's broken between bases, which causes the DNA to unwind. Only one strand of DNA acts as a template. Free-floating RNA nucleotides align to complementary bases via complementary base pairing. Phosphodiester bonds join adjacent nucleotides via RNA polymerase. And splicing of pre-mRNA to remove introns. You could add a little bit extra there about how it becomes pre-mRNA, since you now have a copy of the DNA molecule. But let's go on to Physics Math Tutor and see what they have to say about this. All my homies love Physics Math Tutor. It's not the best resource, I'll be real, like it's a bit... They do get some like very minor things, like a little bit wrong. But hey, look, all my homies love them. It is what it is. Right. So, hydrogen bonds between DNA bases break. I think I put that there. Hydrogen bonds broken between bases. Only one DNA strand acts as a template. 2 marks. RNA nucleotides align by complementary base pairing. So I said RNA nucleotides align to complementary base. RNA polymerase joins nucleotides. Oh, it joins. Hold on. Yep, I said join. So you can't say forms, it joins. And by phosphodiester bonds. And then it doesn't mention it here. Oh, it does. Pre-mRNA spliced to remove introns. So that right there, 1, 2, 3, 4, 5, 6. If that was a 6-mark question, I'm like confident that I could get 6 marks on that. Bear in mind that took me like 5 minutes while narrating it. So it could have taken me even less time in an exam. Bear in mind, I have to write big here. I can write small on the exam. So there you go, guys. All my homies love transcription and translation. Let's do translation now. So in translation, the first thing that's going to happen is we're going to write the name. so I can take screenshots for the VOD. Okay, translation. What happens in translation? Well, what we have is, I like to draw like a burger. This just helps me like visualize it, I think. All my homies love the burger metaphor. So what happens? Imagine this is the ribosome. Even though it's clearly not. So what's going to happen is you are going to have... Oh, I really need new whiteboard pens. What you're going to have is, firstly, mRNA is going to attach to ribosome. You have to mention the ribosome because it's the most important feature of translation. From there, the first codon, or first three bases, codon, is read. You know what, actually, I don't know if that's right. I don't know if you would get a mark for that. You know what we'll say instead? We'll say tRNA brings specific amino acid. We're then going to say... Okay, maybe we should save it. You know what, we'll say it, just see what happens. First, three bases, codon, red. tRNA brings specific amino acid to ribosome. But I guess that's kind of already implied. So that's the first bit. The third bit is tRNA anticodon is going to bind to the mRNA codon. Bro, I'm going to be so pissed if this doesn't come up in an exam. I've memorized this mark scheme. tRNA anticodon binds to the mRNA anticodon. This repeats with new base, new codon being read. You're going to say amino acids keep being brought, joined together via peptide bonds. Amino acids joined together via peptide bonds to form polypeptide. Let's check. So, mRNA attaches to ribosome. 1 mark. Okay, I always get the order mixed up. Firstly, tRNA anticodon and codon bind, and then it brings a complementary amino acid. Amino acid joined by peptide bonds. I did mention that down there. I'm not sure if that'd be a mark. Via ATP. So peptide bonds form. Amino acids. So peptide bonds form via ATP between amino acids. Okay, so really we could write this to say, mRNA attaches to the ribosome, tRNA anticodon and the mRNA codon bind because they're complementary. tRNA brings the specific amino acid to the ribosome, and peptide bonds form between amino acids via ATP. Forming peptide bonds, and then eventually the tRNA is released. Okay, so probably wouldn't have gotten 5 marks on that, even though that was worth more marks. Bro, my neighbours try not to yell and like... I don't know, bro. All my homies hurt my neighbours. Okay. So, there you go for transcription and translation. I'm confident that if a 5 marker came up on transcription, I would get all marks. Translation, I feel like I could easily miss out on 1 or 2 marks. Like, you need to remember that ATP joins together the peptide bonds. You need to remember the start and stop codon, things like that. Just some like basic things which you could easily forget. So that's transcription and translation, which could again be worth in an exam up to 11 marks, maybe 12. That would be lovely. I mean, if you get a question which is related to transcription and translation, remember, buzzword central. Just spam, the same buzzwords. Complementary base pairing, hydrogen bonds breaking, just generic yap. Let's go to meiosis, because I'll be real with you guys, I still don't understand this, but I think I finally picked up on it today, thanks in part to like a little diagram that I discovered. So neighbours try not to repeat the same two words challenge while screaming at full volume. Okay, so this is the situation, right? I don't know the difference between chromosomes, chromatids, homologous, but I can, if you give me enough time in an exam, I will be able to remember it. It's annoying, it just doesn't process for me. So what we have, let's assume that there are 3 pairs of chromosomes here, right? You got, in fact, no, we'll say 4 for convenience. So there are 4 pairs of chromosomes here, okay? Remember your maternal and paternal, okay? Then you are going to have 8 pairs after DNA replication. Just like that. So 8 pairs. Then, during meiosis 1, it's going to revert back to 4 because it's split. And then during meiosis 2, it's going to be like this. I hope that helps you guys, because that really helped me. So 4 pairs, 8 pairs, 4 pairs, and then no pairs, just 4 chromosomes. I guess 4 individual. And in case you're wondering why it's individual, the whole idea here is that they line up on each in the middle, which means that they get pulled away in different orders. Remember that during metaphase 1, so during prophase 1, so during prophase 1 and metaphase 1, this is where your important actions happen. In prophase 1, you have your whole crossing over and independent. So basically, independent segregation is literally where during metaphase, they will line up whichever way that they want. They will line up whichever way that they want, so that when they are pulled, it's a different variation. They assault randomly. That's the whole idea. Honestly, once you kind of understand the whole metaphase thing, it becomes a lot easier. It's taken me up until today to know it, to be honest. Crossing over is a little more complex. It's where your bivalents form between homologous chromosomes, and genetic information is exchanged because a chiasmata forms, which allows the genetic information to be exchanged, because a part of a chromosome's breaking off and swapping. So honestly, I hope that helped you guys, because that helped me so much to understand. 4 pairs, then 8 pairs, then 4 pairs, then 4 individuals. So if you get asked on an exam question to identify the stages of meiosis, I hope that helps. Obviously in humans, it's going to be what? 20... Is it 26 or 28 pairs? 26, I believe. But yeah, that's basically it. I mean, again, this was my weakest topic up until basically today, when now I kind of understand things a lot better and I'm a lot more confident with it. So again, proof that literally like an hour of revision can completely change your mind. Again, I despise this topic up until now. Oh, I'll just be a sec. So yeah, again, proof revision can really help. So what we've done so far is we've gone over not independent segregation. We've gone over meiosis. We've gone over transcription, translation. We do not need to go over the immune response. I swear down, if I have to go over that again, I will end everything. We don't need to go over the stomata opening and closing. Let's go through active loading. Actually, let's go over firstly the xylem. All my homies love the xylem, but it is a little bit complex. Okay, and it is a little bit complex. So let's assume that we have like a leaf. Okay, that's the leaf. And it has like a load of, I'm drawing this in a really bizarre way, but this is to convey the structure. Okay, so imagine that that's a leaf. Okay. And this is the xylem. Again, inaccurate, but come on guys, it's literally a diagram, come on. I guess, okay, well that doesn't look good, does it? So, the whole idea you need to understand is water is cohesive. And this is because H2O, so what you have is you have your oxygen, and then you have your hydrogen, and this obviously, because hydrogen can bond to other hydrogen, via obviously hydrogen bonds are the key component here. Hydrogen bonds allow for cohesion between water molecules, and what this creates is tension, which allows water to be pulled up in a column. So basically what you need to do is, if in an exam you're asked why does water move in a column, it's because of its cohesive nature via the hydrogen bonds. If you're asked, how does water get pulled up, that's when you refer to tension. Because basically, if water is used, then obviously it's going to create a lower water potential. So water is going to move up the column and into the cell. And I guess you could say via osmosis. I haven't actually ever seen an exam question asking you about this, so let me go quickly try find it. Let's try and find it, shall we? Yeah, water evaporates or is transpired for 1 mark. Water potential gradient lowers or lower water potential, which creates tension, which pulls the water up a column. So, because hydrogen bonds are cohesive to maintain the column. Okay, so again, very easy. Literally 1 mark, water potential decrease due to evaporation or transpiration. So in return, obviously since that lower water potential, water is going to move into the cell in a column. Again, very easy. So yeah, a water potential gradient is created, which creates tension, creates a tension to pull up the water. Let's go to, we don't really need to go through active loading. And to be honest, the whole mass flow hypothesis is pretty easy too, honestly. The whole idea is that in the source, because you have all of your sucrose moving out via, I think, facilitated diffusion it'll be. What you're going to have is a lower water potential here, which means water is going to move in via the xylem via osmosis. This creates a high surge of pressure, and as a result, a pressure gradient. And in the sink, it's going to move in because there's a lower water potential overall. It gets used. Oh, and mass flow hypothesis. The whole idea that ringing is used, and that suggests that the phloem is used to transport, it would be, I guess, sugars, the components of photosynthesis, the products of photosynthesis. Because if we have this big tree branch here, and we cut a ring in it here, then everything around here would begin to kind of die, and you would have a massive growth of things here because they're desperately trying to, grow again. So yeah. And it also creates a bulge of water. Radioactively labeled carbon. Okay, so one more thing is for carbon tracing. Basically, the radioactive carbon is obviously used as a product of photosynthesis, so it becomes sugar. This sugar is then transported by the phloem, which means it will go to the sink. So yeah, radioactive labeled carbon, mass flow in the phloem throughout the plants. Yeah. Okay, so this is a quick explanation of mass flow. So in the leaf, sugars are actively transported into the phloem. So my bad, it's active transport via companion cells. So missed out on 2 marks there. This lowers the water potential in the sieve tube cells. We've said that. So water enters by osmosis, which increases pressure, which causes a movement of mass down the pressure gradients towards the sink, and the sugars are used by the sink. So yeah, basically, if you can link it together, so active transport of sugars into the sieve tube cells via companion cells, then you've got everything. So we've now gone through a load of the complex processes. Do we have time to go over digestion? I think we do. Lipid digestion. All my homies hate lipid digestion. I'm looking at you there, Alex. But honestly, I don't blame you. Lipid digestion sucks. Okay, so let's understand something. This is a big ass fat. And I'm not referring to Ashton. I know he doesn't watch these, so he won't care. So we have this fat, okay? What you're going to have is action of bile. Emulsification via bile. And what's going to happen is this fat is going to be broken up, increasing the surface area for a faster rate of hydrolysis by lipase. Lipase is going to break down these lipids into fatty acids and monoglycerides. From there, the bile salts are going to associate to the monoglycerides, and as a result, we have our micelles. These micelles are water soluble, and as a result they increase the solubility of the monoglycerides and basically bring them into the cells lining the epithelium via diffusion. From there, they are going to be reformed in the smooth endoplasmic reticulum into lipids, or I guess triglycerides. And from there, those triglycerides via the Golgi apparatus are going to be placed in a vesicle. They might be modified into like something else, but via the Golgi, the vesicles, they're going to be released into the bloodstream via exocytosis. That's basically it. Emulsification by bile, action by lipase breaks them down into monoglycerides. Let's go check a mark scheme there to make sure we haven't missed out, okay? To be careful, fearful, to be wanting. Okay, so my cells contain bile salts, make fatty acids more soluble, or they bring fatty acids into the cell lining, the ileum. Did I say epithelial? I meant ileum. Fatty acids are absorbed by diffusion. Okay, so they bring it in, but they're actually absorbed by diffusion. So you have to mention diffusion there. Cool. Triglycerides reformed in cells, or chiromicrons, that's a word I forgot. The vesicles, or exocytosis occurs. Cool. Co-transport, you need to talk about this idea of sodium ions generating a concentration gradient which allows sodium ions to move in by facilitated diffusion, or basically, saying sodium ions move in by co-transport along with glucose. And when you're referring to triglycerides, you should always mention the three buzzwords: the action of lipase, the hydrolysis of ester bonds, or the digestion of ester bonds. Again, if a standard deviation, that droplets increase surface area for lipase, so faster action of hydrolysis, my cells carry fatty acids and glycerol through membrane to be absorbed. Reject, my cells increase surface area. Okay. The Golgi modifies the triglycerides, combines them with proteins. Okay. So there you go. There you go, guys. I'm basically, I have no idea what else to talk about. Tell them to bring out the whole ocean, you know what I mean? Don't do that. Don't give us awful questions, AQO, please. I mean, look, I always bring it back to this. Guys. A little bit of reassuring for the day, okay? You could literally get, if you got 40 marks per paper, you're probably going to get a B. Maybe, if you get good RNG, maybe you'll get an A. Okay? Genuinely, check my spreadsheet. If you don't have access, send me an email or something. Check my spreadsheet. I kid you not, I am not crazy. I kid you not, you will literally get, like, so much clarity from it, okay? Look, you need roughly an A over the last, 8 years that AQA have done AS biology. An A is roughly 55%. Okay? What that means is you need about 41 marks per paper. If you really want to guarantee it, because it could, a year before it was 58 in 2023, so it was at an all-time high last year, so just telling you guys that in advance, in case the grey boundaries are wank. But a B is typically, let me just go check my timetable, because I want to make sure you guys don't misinformed here. Okay, look. A B is typically 47. again, there have been years where it's been a little bit higher. 2022, it was an all-time high. 2022, it was at an all-time low. You only needed 50% for an A. 2023 was at an all-time high where you needed about 59%. So just bear that in mind. It varies. There's a possibly 10% here, standard deviation. So yeah, just bear that in mind, guys. Just bear that in mind. If you got, let's even say you got 35 marks on this paper, you probably need about 48. Okay, you probably need about 48 for this one. So 48 marks, is that doable? That's just about 66%. Is that doable for you guys? Do you think you could get 60% of this exam? Let me know, because, I mean, look, we get a question about protein synthesis, you could probably get it. Meiosis, you might not. It's a difficult subject. I don't blame you. I might not. A question about digestion, you've got it. I mean, come on. It's lovely. Question about ecology. All my homies hate ecology. No idea. Question about infection and response. We've got it. We've got it. Guys, seriously, man, good luck, guys, with this. And also, just thank you, guys, because for those who don't know, I don't do these streams to teach people, not like that. I do it because I find if I'm able to explain something, I can revise it much more efficiently. And I mean, yeah, I know that these streams, back in the GCSE days, we would get dozens of viewers because people were all doing the same exams. Now the only people who would ever watch these streams are Alex and Faith, and I don't think Faith has watched one since that proteins question back in like November, and I don't think Alex has watched one in like a decade. So yeah, much thing to say that while I know people don't watch these anymore, and while I do completely understand, you all have different ways of revising. I know for a fact watching a stream for an hour probably isn't your best, way to revise. For me it is, and that's why I gotta thank you guys. Good luck with the exam. Remember this, okay? Look, in that exam, I genuinely think I maybe got, in the last AS one, I probably got about 40%. Sorry, 40 marks. I probably got about 40 marks. My logic was I probably didn't get many questions in the big 5 marker. I probably dropped a couple for that ecology question. I probably dropped 1 or 2 in like the virus question. I deducted marks all around to try and make it as strict as possible. And I came out with about 40 marks, maybe 45, best case. What that means is I need to get another 40 marks. Is it doable? I mean, look, if 10 marks were available for protein synthesis, I gotta hope. I gotta hope. All right, good luck. Oh my God, I can't speak. One sec. All right, so good luck.